This conference will now be recorded.
Okay, let's go ahead and get started. We will probably finish most of Chapter 9 today. And then on Friday, we will come back and do a little bit of gross anatomy here. Just kind of a brief overview of gross muscle anatomy. Again, largely we spend that time in lab working on that, but we'll go through some of it here as well. I will also post a study guide for Monday's exam, so that will be up today, assuming I can actually remember to do it. If I don't get it up today by like this afternoon, so you can send me an email and I'll put it up. It's entirely possible that I might forget again. So there'll be a study guide for that. I will also post at the same time back in, oh God, when I first got here, so 2014, 15, somewhere in that neighborhood, 2014, I think. I made a series of videos with a instructor that used to be here, going through the cat dissection in all of its glory, so to speak. And we have a playlist of videos on YouTube of those individual dissections. So I will also post those to the Canvas site. Students often find them useful not only before the lab, but also kind of later on as they're studying, because we go through and we point out all of the muscles on the cat we dissected at the time. So it can be a useful study aid too. So that will also go up on the Canvas site today when I post the study guide to it. Where we had left off on Monday, we'd been talking about resistance, aerobic exercise, right? Those things that we do to ourselves, basically the damage we would apply to ourselves in order to adapt them to what we're going to be asking them to do, right? So exercise, yes, but also your daily life, right? The thing about it, and this is where all of these kind of vaguely ludicrous motivational things like no pain, no gain come from, is the idea that in order to stimulate development of muscle in one of those directions or the other, you have to demonstrate to that muscle that it is not up to snuff. And the way that we're going to do that, you can't just send it a memo, right? The way that we're going to do that is by physically working it until it breaks. Right? This is what's called the overload principle. Right? And again, like we've talked about before, if you've worked a muscle hard enough that it hurts, that afternoon or that night or the next day, right? It's because of this, right? This is your body telling you, hey, you damaged this, and in terms of gains, right? That's a positive thing for you because you know that you have met and exceeded this overload threshold, and that the muscle itself will be rebuilding itself back stronger again than it was before. Now, little bits at a time, right? But you're not going to go to the gym, lift some weights one day, and wake up tomorrow and you're, Arnold Schwarzenegger. But little bits at a time. This is how this works. Now, the other side of that, okay, is that if you want muscles to be functional and to develop, you have to allow them time to rebuild. Okay, you can't go and do lift weights 7 days a week and expect results. That's actually going to be preventing you from getting what you want, right? You have to give break periods or rest periods in between events of overload for proper muscle development. Right? And again, this is both on the aerobic and the resistance exercise training side of things, right? So any decent exercise program that you put together, you have to put together for you, is going to build in these rest periods, right? So like one day, right, you'll work one group of muscles with resistance exercise. The next day will be aerobic exercise. The next day will be a different muscle group or a different part of the body with resistance again, and then back to aerobic, right? You really, really don't want to push any farther than you can. And that might seem counterintuitive, right? People want results now, now. But results are going to come if you treat them in the best possible kind of way, which is to say, to give them plenty of time to rebuild and to rest in between. I know, and I say this, and I, generally speaking, I bring this up only because typically the classes that I teach at this level are typically mostly women, and typically when I talk about this with women, they don't like the idea of resistance exercise. But I will tell you that if you are looking at a exercise program, resistance exercises are a key part of it. You don't have to lift huge amounts of weight. They can be small amounts of weight if you don't. I mean, it's harder than you might imagine to get massively bulky, right? It's probably not going to happen. And a lot of people shy away from it, just as on the other side of the spectrum, a lot of men tend to shy away from the aerobic side of it for whatever reason. Whatever it is, if you're going to look at being in shape, you have to kind of bring both components in there. It's kind of this weird thing that is thankfully getting less prevalent. The more I talk about exercise programs with people, more people are kind of, yeah, I do a little bit of both. And so that thankfully is going away. It's this really weird old stereotype. I don't know where it's come from, but trying to disabuse the idea to everybody that you should do both. The other side, right, of working out is not working out. And if you don't use muscle, remember that it is expensive, right? So it's like, you go home and you've got Netflix, and you've got Hulu, and you've got Disney Plus, and you've got Peacock, and you've got whatever other YouTube Premium, right? And all these streaming programs you're paying for, right? The reality of it is, you come home every night and you do the same thing. You watch reruns of The Office all day until you fall asleep. You're paying for stuff that you're not using. This is not sound financial sense, right? Same thing here with your muscles. They're really expensive. They burn a huge amount of calories, right, to maintain. So if you're not going to use them, your body's like, no, we're not doing this, right? And you will lose them, right? And this is what disuse atrophy is. If you are bedridden, right? This isn't like, oh, I'm taking a week off and I'm being lazy and I'm sitting around the house all the time. That's not disuse in that sense. It's disuse is in like you are bedridden. Someone has to come in and turn you so you don't get bed sores kind of disuse, right? But under those conditions, right, or if it's in a cast, for example, under those conditions, you will lose maybe 5% strength, right, tensile capacity per day, which is a lot. You lose it pretty fast. You gain it pretty slow, but you lose it pretty fast. And where the big issue comes from is from people that have had spinals, people that have full or partial paralyzation. Even if you are laying in bed all day, every day, you're bedridden, you're still experiencing muscle tone. Right? You are still cycling through those motor units. Okay. If you have a paralysis, right, you're not getting any of that. There is no neural stimulation whatsoever to those muscles. And eventually they will wither to the point where they cannot physically be rebuilt, right? If you have a cast on for a month or two, right, you're going to come out and your arm or your leg, whatever, is going to be pretty weak, right, because you haven't used it. But you can regenerate that, you can rebuild that, you can come back to normal. There is basically a threshold here. When you lose 75% of the initial muscle mass, at that point, it's done. Okay, so one of the things that people have started to do, and I'd say the last 20 years or so, is people that are paralyzed are getting stim. They're putting a TENS unit on, they're getting some stim to try to maintain that at above that quarter of the initial size, enough muscle mass so that if the paralysis can be repaired in some way later on, they won't be SOL, right? And that would be pretty lousy, right? You've got a spinal, and then announce one day that they can fix it, and they go in and they fix it, and you're still out of luck because your muscles have withered too far to be functional. So we do try to do that a lot with those kinds of activities for people in those situations, in case they get to a point where they can be repaired. Okay, so that is more or less the package of skeletal muscle, right? We will talk a little bit about cardiac muscle, just a tiny little bit, but for the most part, from here on to the rest of the chapter, we're going to be discussing smooth muscle. The reason we're going to be discussing smooth muscle more than cardiac muscle is that smooth muscle is profoundly different from skeletal muscle, and cardiac muscle really isn't. Okay, smooth muscle is really, 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 really different. And the first way in which smooth muscle is going to be different structurally for us anyway, is that we typically find it in layers, right? With skeletal muscle, right, we have one line of action, one direction of motion that we want to have happen, so we have one muscle that makes it work. Okay. With smooth muscle, what we're doing is we're forming tubes. We're forming basically like your gut tube and blood vessels and things like that. Things that are going to be, yes, storage, but also we need to move things through them. Okay. And the means at which we're going to use to move things through them requires that the contraction occurs in two separate directions, right? Basically orthogonal, 90 degrees to one another. And so we're going to have one layer of muscle that's going to be circular, that's going to pinch the tube off, okay? And one layer that's going to be longitudinal, that's going to make the tube shorter. By alternating the longitudinal and circular muscles, right, we basically push whatever we need to move along through the tube, like squeezing toothpaste through a tube. So if we look at just kind of a basic cross-section, there can be more, and there sometimes is more than two layers, but there's almost always at least two layers. If we look at, find my cursor again, section here of small intestine, right, we have this inner layer of muscle right here. Okay, that's going to be the circular layer. That's going to be the one that's going to pinch off. When we talk about sphincters throughout the digestive system and elsewhere in the body, right, something that's going to close off a tube, it's circular muscle. Okay, or the sphincter pupillae, circular muscle. Okay, same basic idea. The outer layer, right, the one out here, is the longitudinal layer. That's going to be the one that makes the whole tube shorter. Okay, so these muscles alternate contraction to move food through them, or whatever else needs to be moved through them, I guess. Now, if we look at them in longitudinal view, right, we see that the cells themselves are not running the length of the muscle. They're not particularly long or large in really any way, shape, or form. And they lack the striations that we associate with cardiac and skeletal muscle. They're really, 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 really different. Unlike skeletal muscle, they're uninucleate, right? They have only one nucleus rather than multiple nuclei. They are mitotic, so they can divide. So if you get stabbed in the stomach, you've gone to a Raiders game or something, you got stabbed in the stomach. While the skeletal muscle, right, if your abdominal muscles won't regenerate, the muscles of your small intestine will, right, or the stomach will, I suppose, if you get stabbed in the stomach. So that's kind of a neat, usable thing for us. They are not packaged in the same way that skeletal muscles are. We don't have fascicles. We don't have perimysium. We don't have an epimysium. We have only basically the cell membrane, the endomysium. Okay. Internally, right, we don't have the striations, so we don't have myofibrils. We don't have sarcomeres. We don't really have anything that would be described as a triad, generally because we more or less lack a sarcoplasmic reticulum. There is a sarcoplasmic reticulum, but it's really poorly developed. Okay, we are still going to use calcium as our molecular kind of trigger between excitation and contraction. Right, the coupler there still is calcium, but that calcium is going to come from a different place. Okay, that calcium is going to come from is little pouches on the outside of the cell, right? So rather than having T tubules go all the way through, we used to have little pouches on the outside. These are called caveolae, or little caves. Okay, and these caveolae are basically going to be places where we can kind of slurp up calcium from the extracellular fluid. Calcium just kind of hangs out in there, and when we need it, we pinch those off, we form little vacuoles, and we release it into the sarcoplasm from there. So we arrive at calcium from a different source. Then again, of course, right, this notion of this layering, right? We get into like the stomach, we have actually 3 layers of muscle, but generally speaking, we have these 2 here, the longitudinal and the circular layer. And again, alternating, shortening, pinching, shortening, pinching, shortening, pinching, shortening, pinching. What this does for us, right, this process is actually called peristalsis, right? Where again, it's kind of like, and I say this, I had a roommate in college that didn't do this, and I to this day, I'm angry about it. But if you have a tube of toothpaste, right, you start at the bottom, right, where the little fold is, and you move upward towards the nozzle, right? You pinch and you slide, right? And the toothpaste comes out the nozzle at the other end. My roommate always started at the nozzle end, and it really pissed me off. It was just such a waste of toothpaste. He actually blew out a toothpaste tube one time doing that. Like the other end unfolded and toothpaste went everywhere. It's a rental. It's really annoying. Anyway, that's basically what peristalsis is, right? The circular layer is the pinching part, okay, and the longitudinal layer, right, mimics the sort of the sliding action that you're doing there by shortening that tube in that region, and it pushes that food along, right, or whatever else we're talking about. And then we relax that, and we pinch again, and we shorten, and we pinch and we shorten, we pinch and we shorten. Right? And we kind of stepwise move food or whatever is being moved through the tube, okay, through the alternating action of these 2 muscles. Again, generally, we think about it in terms of the GI tract. So the bolus or the chyme or the feces, wherever step we're at, okay, but it can be other things as well. Okay. So again, just by way of comparison here, right, we'll kind of ignore cardiac muscle here in the middle just for the moment. Our old friend skeletal muscles, right? If we then look over here at smooth muscle, they're really kind of alien, right? They really don't have anything recognizable in terms of being a muscle cell compared to something like a skeletal muscle. And again, that's down to the fact that they lack, right, this internal structure that we're typically looking for in a contractile tissue here, right? These myofibrils, something that we see in cardiac and skeletal muscle mouth. They lack those things altogether, and indeed lack any sort of tendon, lack any sort of peri or epimysium. Just really, really very different. Another thing that's going to be kind of unusual, I guess, maybe is the way to put it, coming from a skeletal muscle perspective, is how we actually interact with the nervous system. Now, again, the smooth muscle in general is oftentimes perfectly happy having nothing to do with the nervous system whatsoever. We have a number of parts of our smooth muscles are what we call autorhythmic, right? They generate their own action potentials on a molecular clock, basically, or in response to stretch. That's another really common way in which smooth muscle stimulates itself to contract. But we do, in some cases, have kind of an oblique connection with the nervous system. Okay, and rather than having a typical neural junction, right, a typical synapse, where we have an axon terminal, we have a very small gap between that, we have the synaptic cleft between that and the adjacent cell, and one terminal to one cell kind of a deal. We have none of that. Okay, what we have instead is a situation where the axon itself has swellings. Right? The axon branches and has all these little swellings called varicosities all the way through it. Varicosity, right, comes from the same root that gives us varicose veins, for example. Varicose veins, if you're unfamiliar with what's happening there, just through the sort of normal action of gravity, right, we have blood kind of pooling against valves in major veins. As it pools, as you stand for a long time or whatever, it tends to stretch that vessel out, and we get these swellings above the valve and veins, varicose veins. Same idea here. Varicosity is just a swelling. And those varicosities are full of neurotransmitter, but they don't directly contact, right, or even come close to any particular individual cell. What they're going to do is they're going to release neurotransmitter kind of like, this is horrible, but this is the best analogy I've ever heard. Fish spawning. I don't know if you know how fish reproduce, but they kind of swim in the neighborhood of each other, and then one releases sperm and the other releases eggs in big clouds, and we hope for the best, right? This is kind of the same thing. We're just kind of releasing a cloud of neurotransmitter, and we're hoping for the best. Okay? This is called a diffuse junction rather than like a normal sort of synapse kind of junction. And if we look kind of at how they're arranged, right? So here's our nerve, right, from the autonomic nervous system, a branch from the autonomic here. So sympathetic, parasympathetic, doesn't really matter. With these little varicosities kind of scattered all the way through it. And you see they're kind of nearby the smooth muscle cells, but they're not making any real contact. And in fact, most of the muscle cells in this package of muscle are not anywhere near it, right? They're not going to be anywhere near that at all. So it's very, very different in terms of the interaction that we're going to have there. Now there are cell junctional type issues here. We've got a bunch of gap junctions that connect all of these cells. So functionally it doesn't matter as long as one of these cells gets neurotransmitter, because they're all connected by gap junctions. They will all contract roughly simultaneously. But it's just a different mechanism of doing it. Now, how do we actually go about contracting? If we don't have biofibrils, don't have sarcomeres, how are we going to do this? Well, we do still have thick and thin filaments. Okay. Ratios are a little bit different, right? There are significantly fewer thick filaments than there are in skeletal muscle, but it doesn't really matter, right? The big primary difference here in terms of structure is how they're arranged, right? So in skeletal muscles and cardiac muscles, they're all arranged in the same direction, right? They're all in one big line, right? And so when they shorten, right, the whole muscle shortens in one direction and one direction only. We don't go off on crazy tangents and kind of bend. We get from here to there. That's the extent of what we can do. Okay. With smooth muscle, they're not arranged in one big line. They're not all lined up. Okay, they crisscross. They attach at various points along the inside of the cell, so that rather than contracting and getting shorter in one dimension, they kind of corkscrew. And so they do get shorter, right? But the arrangement of the filaments means that they kind of get shorter in a lot of directions at once, right? They don't kind of have this linear action like we expect to find. The thin filaments themselves are also slightly different in terms of their arrangement. We lack troponin, tropomyosin, okay, that complex there, that's not here. What we have instead is a enzyme called calmodulin. Calmodulin is actually going to bind to calcium, and that's going to be what's going to be our trigger. We'll talk a little bit about that in a minute. The arrangement of these things, because they're not going to one end of the cell, basically connecting to connective tissue on one end and connective tissue on the other, like we see in a skeletal muscle. The danger, of course, is always sort of ripping off the inside of the cell, right, or damaging the cell wall or what have you. So as these filaments attach to the cell membrane on the inside, they actually attach to these relatively little thick plaques called dense bodies that are just a little bit more secure. They're a little bit kind of more of an anchor for these filaments, so that when they pull against it, we're not really running the risk of breaking, basically, to make that happen. And these are essentially the Z discs. This is basically what these are. When we talk about sarcomere shortening, right, we're pulling on those Z discs. Here we're pulling on these dense bodies. Right, that's basically the idea. Okay, so if we look at kind of a cartoon here of a smooth muscle cell, just one cell, a couple of things to point out here, right? So here in the sarcolemma, right, the cell membrane, we've got all these little pockets. Right? These are the calveolae. These are where we are storing calcium from the outside of the cell. Okay. Nucleus in the middle. You've got all these little intermediate filaments. These are kind of our little anchors attached to these dense bodies, right? And what's going to happen is when we contract, we're going to kind of tug on all of those dense bodies, and we're going to corkscrew. We're not just going to get short. We're going to kind of twist and corkscrew and kind of get smaller in multiple dimensions because we're not pulling on one end of the cell and the other. We're pulling on those dense bodies and pulling them closer to one another. Now, the actual contraction here is, I would say, significantly slower than what we would be used to with skeletal muscle. The idea here is not, you're not using your colon to run away from a bear, right? Presumably. Much lower stakes kinds of issues here, right? So we're not going to be concerned with speed so much as we're going to be concerned with coordination, right? So think about it like, when you learn to play a musical instrument, right? You learn to play guitar or play piano or whatever you're learning to play. You don't come in and they don't hand you a piece of music and say, play this as fast as you can, right? You play it slowly, right? Especially at first, right? Because the important part is the right notes in the right order, the right time, right? With smooth muscle, right, the important things here is that we don't get things out of sequence. If things get out of sequence, we don't do our job, right? If you don't alternate squeeze, shorten, squeeze, shorten, squeeze, shorten, we don't move things through the tube, right? So we go slow to make sure they happen properly. Okay. Now again, we don't have every cell wired up, right, to a neuron. So the way that we make this coordination happen is we form what's called a functional syncytium, right? SYNCYTIUM, which basically is just a fancy way of saying we take a bunch of cells and we functionally make them one cell, right? We hook them all together with gap junctions. The sarcoplasm is continuous between all of the cells in this group here, so that if we get one reaction, one electrical signal in one cell, they all get it roughly at the speed of electricity, which is more or less instantaneous. Okay. So we don't have to have that wiring kind of elaborate setup that we see in skeletal muscles because we have gap junctions instead, right? They're what's going to make the thing behave as one big unit rather than elaborate wiring as we see in other things. The filaments here, actin-myosin, sliding filament still holds true. We still form cross bridges. All those sorts of things continue to happen. Okay. One big fundamental difference here that I want to point out that I think is important for you guys to know and to remember. Remember, with skeletal muscle, right, we use ATP to relax. Right? We use ATP to pull back the myosin head away from the thin filament. We use ATP to pump calcium into the sarcoplasmic reticulum. Right? Here with smooth muscle, the contraction is energized by calcium, by ATP. Okay. So here in smooth muscle, we use ATP to contract. Skeletal muscle, we use it to relax. So that's one big fundamental difference here. Now, where things get interesting is with the role of calcium here. So binding of calcium to calmodulin activates calmodulin. And calmodulin is what we call a kinase. A kinase is something that takes a phosphorus molecule or phosphate molecule and attaches it to something else, right? It forms a phosphate bond. It gives something else energy. Okay. So our ultimate chain here is this calmodulin is going to basically interact here with the myosin heads and allow the myosin heads to move. We're going to phosphorylate the myosin. Okay. We're giving the myosin energy. So rather than exposing active sites like we see with skeletal muscles here, basically what we're doing is we are providing energy to the thick filament to form cross bridges, to shorten, to pull things. So the calmodulin itself is actually not in contact with the thin filament at all. It's oftentimes nowhere near it. Okay. It's a very, very different system. We're still using calcium as our trigger. We're still forming all the cross bridges type stuff, but again, we're going about it in a different way. The fundamentals of how we're using the machinery are different. And the machinery might be the same, but we're using it in a very different way. So if we want to kind of see this in visual form here, right? If you have the Mary Ben Home textbook, this figures in there. If you have the OpenStax book, there's a similar diagram to this. But basically, this is going to be our process, right? For contraction here, at least for excitation contraction coupling, right? In smooth muscle. So we're going to begin by bringing in calcium. Again, typically, right, we bring this in from the outside. We don't have much sarcoplasmic reticulum. We do have some, but it's really not a major component. Most of this calcium comes in through the caveolae from the outside, right? Again, the important thing here is that our trigger is calcium in the sarcoplasm, same as we saw before. That's not any different. Difference here, right, again, is what that calcium binds to. In this case, we're binding to calmodulin, or activating calmodulin. Okay? We're basically setting off a fairly lengthy chain here of kinases. Again, with a kinase, all this is it's just handing energy from one molecule to another. That's all a kinase does. If you want to think about it like that, that's fine. Okay? Ultimately, right, this kinase chain is going to end by giving ATP to the myosin, okay, and by attaching phosphates and the phosphate bonds and the energy to the myosin heads. So the myosin heads can form cross bridges. This is the exact opposite of what we've seen with skeletal muscle. We bind ATP to myosin to pull them back, skeletal muscle. We bind ATP to them to form cross bridges in smooth muscle. Once this happens, right, we kind of continuously cycle through this. We phosphorylate, we dephosphorylate, we phosphorylate, we dephosphorylate, form these cross bridges, and we walk thin filaments along with thick filaments, just like we do in skeletal muscle. One minor difference, I guess I would say, between those two, in skeletal muscle, thin filaments get pulled in one direction only, towards the middle. In smooth muscle, we have oftentimes one thin filament being pulled one direction and the other thin filament being pulled the other direction. It doesn't really matter. Just kind of a quirk of how these things are arranged. So, I guess I think it's an interesting system. It's certainly a different system than we're used to, but again, it uses a lot of the same familiar parts to it, so it's not totally alien. Right? And that's our contraction, excitation, contraction coupling. Now, the contraction part of it, again, is functionally a little bit different here. We're forming these cross bridges. We're walking our filaments together. We're pulling the dense bodies together. It's not a fast process, right? They contract very slowly. They relax very slowly. But the upshot of that is that energetically, it's significantly lower cost, right? You can hold a contraction with smooth muscle for a very, very long time, right? With comparatively little ATP cost to it, compared to, say, a skeletal muscle. They're really, really quite efficient in that regard. And something else that we often see that helps this is that the myofilaments themselves can, during the cross bridge cycle, can actually just kind of, if we're going to be holding a contraction, will just latch together. And they won't continue the cycle, they'll just latch together and bind together and basically kind of do what we see with rigor mortis, right? They just form the cross bridges and hold on to them. And again, that's a very energetically efficient way to do things. If we're going to go and relax that smooth muscle, Okay, we're going to do the exact same thing we do with skeletal muscle. We're going to get that calcium out of there. Right. Here, our primary means of getting that out is to get it out of the cell altogether. Right, we pump it back into the extracellular fluid, into the interstitial fluid. We do put some, again, in the SR, but again, mostly we're talking about pumping it out of the cell altogether. In terms of how we regulate and induce these contractions, we've got a number of different mechanisms by which we can do that. We can do it through nervous stimulation. I've already seen some of that already. We've got hormonal triggers that we can talk about, other chemical binding, other ligands that we can talk about here as well. Neuroregulation is pretty straightforward, right? We have a neurotransmitter here. We bind to the sarcolemma of one or more smooth muscle cells. This causes an influx of calcium, right? So rather than generating an action potential, right, having that action potential open sodium channels or open calcium channels, here we have the action of the neurotransmitter binding open the calcium channels directly. And again, we might experience multiple different neurotransmitters here. With skeletal muscle, it's always acetylcholine. When we start getting into smooth muscle, because we're in multiple different parts of the body, sometimes it's acetylcholine, sometimes it's not. It can be a handful of other things. So depending on the neurotransmitter, depending on the receptor, we might get different responses. Hormones, other chemical messengers here. Again, a lot of smooth muscles have no real connection to the nervous system at all, right? And so we might require a contraction and not have a nervous system pathway to get there. And so in those cells, typically, these are where we typically find the sort of the autorhythmic or the pacemaker cells that have an internal chemical clock, okay, based upon the degradation of certain proteins at a given rate. We cycle through contractions and non-contractions. Sometimes, however, we have ways of modulating those through ligands, hormonal or otherwise, typically through G protein link systems. This might be, with the digestive system, we think about primarily as hormones. With the circulatory system, we think about primarily here PCO2, so what's the carbon dioxide content in the blood, but the pH of the blood. Obviously, those two are linked. That will result in vasoconstriction or vasodilation, depending on circumstances. But again, non-nervous system cues that can relate to contraction or relaxation. One of my favorite things about smooth muscle, I think it's really cool, is what we call this stress relaxation response. And my wife, when her grandma was one of the meanest people I ever knew in my whole life. Her grandma always used to yell at her for eating. And when we first started dating, I mean, this was a long time ago, but it was like 9 pounds, right? But she'd always yell at her for eating, right? You're eating too much, you're going to stretch your stomach out, and then it'll never go back, and you'll be hungry forever. Sort of mean shit you would say to somebody, man. That's 1,000% not true. It's unbelievably incorrect. Smooth muscle has the capacity to stretch, right? It is elastic, right? But it doesn't stay stretched, right? So if you go out and eat a big meal, right, and you really fill your stomach to capacity, right, your stomach is going to stretch way out, right, to accommodate that. But it's not going to stay that way, right? When the food moves on into the small intestine, it shrinks back down to its normal size. It doesn't get floppy or anything like that. The other part of it that's important here for us is that if you think back to how skeletal muscles work, right, we have that relatively narrow band of length over which muscles are fully functional, right? About 100 to 120% resting length. And above that and below that, we lose the capacity to generate tension pretty fast. With smooth muscle, we don't have that. We have a stretch, right? And in the immediate aftermath of that stretch, we're weak, right? In the same way that a skeletal muscle would be weak. But we have the ability to reset our resting length for the temporary periods of time, so that your stomach, when filled and stretched out and kind of bloated, right, isn't weak. It is at full strength. It can continue to operate just as it would at sort of a smaller size, right? And then we move on. The stomach shrinks back down again to its normal size, and we reset again, and we go back to our original resting length. So it's a really amazing system. It's a fascinating system, but it's really, really important because when we think about hollow organs, where smooth muscles are, we're going to be experiencing big changes in internal volume, right? From empty to totally full. Your bladder, your stomach, your intestines, your blood vessels, all of these things, right, are going to be experiencing multiple sort of conditions every day. And so we need to be able to be functional at all of those. And that's why this is. Something else really cool we touched on earlier is this notion of being mitotic, being able to divide. Technical term that we're going to apply to this is hyperplasia. Basically, smooth muscles can get bigger by making more cells. So the classic example of this is with the uterus, right? If you're a woman and you're born, right, you have a uterus at birth, but it's very small. You're not using it, right? There's no reason for it to be big. It's just kind of in the way, right? But as you progress and you pass pubescence and biologically, if not emotionally or mentally, prepared to have children at that point, right? At that point, the uterus becomes significantly larger, right? It grows significantly. And it does so by adding more cells. It actually gets bigger by making more muscle cells. The uterus is pretty cool. It's like the strongest muscle in the body. It's massively strong. And it's all smooth muscle, which is pretty cool. And obviously then during pregnancy, we also see changes to the size of the uterus during that as well. Again, based around this hyperplasia, this ability to add more cells. Oh, we've kind of already talked about this. I'm just going to, I guess, put this up here. In both editions of the book, there is again, this kind of chart in there comparing various things. So if you're trying to find like a one-stop, how are these things different kind of thing, this is a good place for that. You could pause the video too, I suppose, if you wanted to. With smooth muscle, we have some instances where some of them are going to behave like they've got motor units, and we've got some where we are not going to see that at all. We're going to describe these as either being unitary or multi-unit, okay? Depending on variation in how the muscles cells are arranged, how they are innervated, if they have innervation at all, what sorts of stimuli they respond to, how they respond to them, that kind of thing. So the first of these categories here, referred to as unitary or smooth, visceral smooth muscle. Visceral here, viscera, right, is typically your organs. This is kind of a catch-all term we use for that. And so we find unitary muscle, unitary smooth muscle, in all of your hollow organs except your heart. And your heart, of course, has cardiac muscle. It's off on its own. And this is, again, that arrangement we see with these opposing sheets, right? Well, one circular layer, one longitudinal layer. We've got varicose junctions or diffuse junctions and varicosities associated with them, so we do have connections to the nervous system, at least subtly. We have either autorhythmic or stretch-induced spontaneous contraction in them also, so they can contract on their own. And because they are so, the importance of coordination here is so important. Importance is important, how about that? Typically, they're connected by gap junctions. They're these functional syncytia, right? They're basically one big cell connected to each other. With multi-unit, we find these predominantly in your airway, so in your respiratory system. They're also associated with the iris of your eye, right? So the smooth muscles that are controlling your pupil size. The erector pili muscles attached to your hair follicles are multi-unit. Some large arteries have them. Elastic arteries, some of the larger muscular arteries have them. And again, connected by gap junctions, but here we find that spontaneous depolarization, so autorhythmicity, etc., not real common. Okay, typically, when we talk about a multi-unit smooth muscle, we're talking about something that's going to have input from the nervous system directly. Again, we think about the pupils, right? Sympathetic and parasympathetic nervous system controlling one muscle or the other, right? They're going to behave a little bit more like skeletal muscles in that regard. Right? We don't see them having a molecular clock. We don't see them responding to stretch in the same way. We basically see them turned on by the nervous system or turned off by the nervous system. Okay, we'll call it good there. We'll come back on Friday, finish off the last little bit here of the chapter, talk about some muscles themselves, kind of as an overview. Again, if don't see a study guide by early this evening, please send me an email and I will make sure I get it out.